General scientific articles. He has filed one patent and he has developed one germplasm. Sir is extensively involved in teaching of both basic and advanced courses in the biochemistry, plant molecular biology and biotechnology to the postgraduate students of NRCPB as well as for IRI, both MSc and PhD students. Sir has been awarded with many awards. To name few, one is the prestigious Lal Bahadur Shastri Young Scientist Award for the biennium 2003-4, which has been awarded by the ICR Ministry of Agriculture and Farmers Welfare Government of India. And he has been also awarded as Young Scientist Award by the Indian Science Congress Association during the year 2000-2001. And he has been also been awarded the DBT Overseas Associateship Award during 2004-5 for his postdoctoral research at Washington State University, USA from DBT. And he has other responsibilities like he is a Joint Secretary for Society of Plant Biochemistry and Biotechnology, he is an Executive Editor of Journal of Plant Biochemistry and Biotechnology, and he was the resource person for 15th Genetic Congress Trust, and he is a member of Selection Committee for HRT Group of Council CSIR. Today we have with us Dr. Ramcharan Bhattacharya sir from NIPP, who will be delivering a lecture on housekeeping genes for expression analysis.
So, very good morning to all of you. I am sure that you must be enjoying the training. There is keen involvement of various faculty members from these institutes, and I can see that Dr. Sagar is with you almost all the time, guiding you through the courses as well as the other time hours. So, today our topic is about the gene expression analysis, and particularly how do you normalize. There is a process called normalization in gene expression analysis that I will come into detail. It is use of housekeeping genes in normalization process while doing the gene expression analysis. So, the lecture will contain few basic things because you are from entomology background. So, I thought always it is good to build up from the basics. So, the entire lecture will contain some idea what do you really mean by gene expression analysis, then how do you do gene expression analysis by using RT-PCR, and then some difference when you do it through PCR or you use qPCR, and then what are the kind of sources of errors or sources of variation that can disturb your interpretation in doing this analysis, and then how do you make use of gene expression, how do you make use of housekeeping genes in normalizing or in nullifying those variations or errors. And also I will touch upon a few statistical algorithms that are popularly used to find out what is a good normalizer, right. And while designing the lecture, I kept in mind the best convenience for you, the most lucidity of the lecture, so that you understand to the fullest possible extent. And in doing so, many of the slides I have accessed from web resources. So, as such there is nothing like copyright. So, many of the slides you can find taken from web resources. So, I acknowledge those who have put those slides in the web resources, and I acknowledge that their sharing with those slides is helping us to popularize the science, to help the students in understanding the subjects. So, first, what do you really mean by gene expression analysis? I am sure that all of you have read about the central dogma of life process. So, what the central dogma says? Central dogma says that in deciding a trait, there is a causal piece of DNA, right, we call it gene. So, that DNA is first transcribed to give rise into a RNA molecule, and the process is called transcription, right. The transcription process, the RNA polymerase, there are many RNA polymerases that take care of transcription process, and then those RNAs, some of those RNAs are mRNAs, and those mRNAs are translated by the process of called protein translation and produce a protein, right? And these proteins can be of diverse type, they could be structural proteins, they could be metabolic enzymes and so on, and all the metabolic processes are run by enzymes. So, ultimately if you see that all metabolic processes are run by enzymes and all enzymes are coded by genes, right. So, you can understand that one can detect a specific RNA to conclude whether a particular gene is expressing or not, because producing RNA is part of gene expression. You agree or not? If a gene has to express, if a gene has to manifest into a trait, then it has to go through the transcription and then sometime translation and so on. So, therefore, one of the methods of studying gene expression has been to detect the specific RNA. If it is there, you say yes, the gene is expressing. If it is not there, you say no, I do not find any expression, right. At the same time, you can do comparative analysis between samples or among the samples to say that a particular gene is expressing more in this sample or that sample or some other sample, right. So, primarily when you do gene expression analysis based on RNA or mRNA, you can do two things. You can detect whether expression is there or not by presence or absence of that specific RNA, or you can do comparative analysis among the samples to say in which sample you find more of the gene expression. So, many of your research will involve where you need to analyze that in which particular plant or under which particular circumstances a gene is expressing more. So, you can always do comparison and conclude on that. Then here we talk about the analysis of gene expression by RT-PCR. RT-PCR, here we mean reverse transcriptase PCR, right. So, I am sure that by now you understand what is reverse transcription process. It is reverse of the transcription process where you produce a complementary DNA or cDNA from a mRNA, and in doing this the job is done by a particular DNA polymerase which is called reverse transcriptase. They are called reverse transcriptase because they do reverse of the transcription process, otherwise they are a kind of DNA polymerase. You might have heard about DNA polymerase. DNA polymerase does DNA synthesis, and primarily what you believe that DNA polymerase does DNA synthesis using a DNA template, it copies. So, the difference between this particular type of DNA polymerase which we call reverse transcriptase is that it uses mRNA as template. Instead of DNA, it uses mRNA as template. That is the only specificity, otherwise it does the same thing. It starts with the existing piece of small primer and then it add on new nucleotides to make a copy of a particular mRNA. We call it cDNA, right. And in laboratory, if you do an experiment involving reverse transcriptase, you will use either of these enzymes which are commercially available. One is called AMV or avian myeloblastosis virus reverse transcriptase, and another is called MMLV, which is reverse transcriptase from Molnoymarin leukemia virus. So, these are commercially available enzymes, and a few more things I just wanted to tell you, because reverse transcriptase is a very important step when you do gene expression analysis by RT-PCR. Remember that this is a very sluggish enzyme. Sluggish enzyme means it is very slow in its action, very slow in doing the DNA synthesis process. So, many a times one of the way of troubleshooting your experiment, suppose your experiment is failing and you are finding that the reverse transcription process is failing. So, remember this point that sometime we increase the time of reverse transcription and we start getting good result, right. So, this enzyme is very slow, and another drawback with this enzyme is that many a times it tend to incorporate errors, but those are different aspects. I will not go into much details of that. And in doing this reverse transcriptase, again like DNA polymerase, essentially you need a primer. It cannot start the copying process de novo. You have to start with a existing small piece of DNA, right, or oligonucleotide, and then the enzyme can add on. The enzyme can add on. So, essentially in doing reverse transcription reaction, you have to take a primer sequence. So, your substrate is here what? Your substrate here is mRNA, right, and your enzyme is reverse transcriptase. So, what are the other things you got to add into the enzyme cocktail? In the reaction cocktail, you have to add a primer, you have to add DNTPs, right, enzyme and the proper buffer for that enzyme. While deciding on the primers, you have few options like say your few options like one can use a gene specific primer. Suppose you know the sequence of the gene, you are trying to analyze the expression of that particular gene. So, since you know the sequence of the gene, you can always derive a gene specific primer, right. So, one can use gene specific primer. Alternatively, if the gene sequence is not known or if the purpose of the experiment is to convert all the mRNAs into their corresponding cDNAs. So, then you can prefer to use a oligo DT primer. Oligo DT because we know that eukaryotic mRNAs, they invariably contain a poly A tail at the 3' end. So, it is a generic in sequence, it is a common in across all the mRNAs. So, knowing that, one can use a oligo DT as a primer, so that it binds to all the mRNA in the isolation and then do the reverse transcription process. And many a times also where you do not bother to produce the complete copy of the mRNA, you can use random hexamers as a primer. So, they are random means and they are hexamer. Hexamer means there will be 6 nucleotides and in all possible combination, ATT, GCC, ATG, GCC, like that, all possible combination. It will be a mixture of oligonucleotides or hexamers. When that is added, so they will find their complementary sequence here and there across the mRNAs at different regions, and wherever they bind, they do synthesis. So, you get a population of small pieces of cDNA, but that will represent your mRNA population. So, that can be used further. So, but in doing RT-PCR, once you do the cDNA synthesis, your next step is PCR. So, RT plus PCR. RT you have done, now you have to do PCR, and I am sure you might have already performed PCR reaction in laboratories, right? They have done. So, you know what are the various components that are needed in performing a PCR. The only difference is here your template is the cDNA that you have prepared in the RT steps. That is the only difference, otherwise it is as good as PCR. So, you add everything, you run the PCR, and what will be your primer? Here your primer will be the gene that you are studying. Understand? So, even if you have done the RT reaction in a generic way, means you have used oligo DT or random hexamer, there is no specificity, all the mRNAs are converted into cDNA, but in doing the PCR you assign the specificity, because there you use a specific primer and then it will amplify a specific cDNA and you will be studying the expression of a specific gene, right. So, you can assign specificity right at the beginning at the RT step. While doing RT, you can use gene specific primer. Also, if that is somehow not possible, or if you do not want to do that, if you want to use that cDNA for other purposes also, so you may prefer to do the PCR step by using gene specific primer and do the RT step using a generic primer. Fine, clear? Okay, very good. So, now PCR amplification, just to refresh your memory, you do how the PCR amplification occurs. You start with certain number of copies of template DNA and then it starts amplifying from 2 to 4, 4 to 8, 8 to 16, like that. So, with every number of cycle it increases. And how do you find the amount of or number of copies produced at the end of a particular number of cycles? The formula is 2 to the power n, right. So, PCR can rapidly amplify and produce millions, billions of copies of a particular piece of DNA or cDNA in our case. Now, this RT-PCR process or RT-PCR reaction, it can be used for many different purposes. It can be used for gene expression study that we will be talking today. It can be used for various diagnostic purpose where detection of a particular mRNA is the diagnosis, right. So, it can be used for diagnostic purpose. It can be used for isolation of intron free coding sequence of a gene, because your starting material is mRNA. It is a processed all the introns have been removed, spliced out, right. So, many a times eukaryotic genes as such, if you try to isolate them from the genomic counterpart, they could be larger or bigger in size, and you may not need that, you need only the actual coding sequence. So, sometime or many a times we prefer to do the RT-PCR to identify or isolate that gene sequence. So, intron free gene sequence. Then you can also do isolation of novel genes based on conserved domain. Suppose this is a bit out of the track, but just to tell you few lines, suppose you do not know the gene sequence, but your strategy or the existing knowledge, literature knowledge allows you to find out the protein sequence, the amino acid sequence of the protein, right. So, starting from the amino acid sequence, one can find out the possible nucleotide sequence, right. So, if you find out a conserved domain or amino acid sequence, from that you can design primers, and using those primers you can isolate the actual gene sequence, right. So, that is again a major use of RT-PCR, but today we will primarily focus on one of its use, that is how do you use RT-PCR for gene expression analysis. So, again I recall that when we say gene expression analysis by RT-PCR, you can primarily do two things: detect presence or absence of expression of a particular gene, and then comparative study. Now, by this time I am sure you might have understood that how do you detect whether there is a presence of a particular mRNA or not. That is very simple. You run the RT-PCR reaction, you do not get any amplification, you say no, there is no expression. You get some amplification, you say yes, there is expression, right. But the interesting part is to make use of this technique in doing comparative study. I give you 5 samples and I ask you that you find out in which sample gene A is expressing at the highest amount, or I ask you to rank this 5 sample in terms of the higher expression of gene A, right. So, there you have to do a comparative study. So, in doing that I take you to the basic process of PCR, how it works. Suppose you are running a PCR. So, how it works? You start with one copy. Suppose your amount of target DNA is just one copy, means one molecule. So, at the end of one cycle it will be 2 copies, at the end of 4th cycle it will be 16 copies, at the end of 8th cycle 2, like that it will go, right. But then if your beginning number of copies is 4 instead of 1, same reaction, but it will have a different kinetics. So, here the kinetics is 2 to 4, 4 to 8, 8 to 16, 16 to 30. But so the assumption is you are starting with only 1. But suppose instead of that you are starting with 4 copies. It will also go in the same way, but the kinetics will be different. Here at the end of first cycle it will be 8, 4 to 8. At the end of another cycle 16. So, at the end of 3 cycle it will be 32, right. So, you see here you reach 32 at the end of 5 cycle in this case, where the number of copies is 1, but you have reached 32 at the end of 1, 2, 3 cycle. Do you understand? So, now if I run a particular number of cycles, say I run 25 number of cycle for both the cases, in one case where it is 1 copy, in another case where it is 2 samples, I do not know, I ran the PCR for 25 cycles. So, what will happen? Here whatever will be the amount of DNA, in the other case it will be 4 times. Do you agree? And if I terminate the reaction at 20 cycle, there also it will be, ratio will be again 4 times more. So, kinetics pattern or the graph will be progressing in the same way, but the end product at any point of time, you terminate at 20 or 25 or 30, wherever you terminate, the amount, the ratio will be representing the ratio that was there in the actual sample. So, that is the whole crux, that is the whole principle. So, I have unknown samples, I ran PCR, same number of cycle, and then I analyze the amount of product that I have got. And if I see that okay, between sample A and sample B, the amount differs by threefold. So, I would say that my beginning number of template was 3 times more here. So, that is fold expression. So, how the amount was more? Your gene is same, but amount was more in one sample, amount was less, or number of copies of mRNA was more in one sample, number of copies of mRNA was 4 times less in another sample. How? It is the same sample, it is the same gene from the same organism, only difference is expression. In one case, it is expressing at 4 times more efficiency. Okay? So, thereby we say that yes, gene is expressing more in this sample, how many fold more? 4 times more, sometimes 3 times more, right. So, in this way one can do the, so as the PCR goes on, the amount of DNA will straight away increase 10, 100,000 like that, you will get this graph, and then you will reach, we call it a plateau. You will reach a certain point where the increase is not in the geometric progression, or increase is not in the logarithmic phase. It is same like microbial growth, you know, if you study microbial growth, it also the same kinetics you get. So, after a certain point you reach. So, the only condition, so you see here, it does not matter when do I stop the reaction. I can stop at 20, I can 20 at the end of 20 cycle, I can stop at the end of 25 cycle, I can stop at the end of 30 cycle, does not matter. But to get the actual ratio of number of copies reflected in my product of PCR, the only important thing is you have to capture within this region, where there is a straight line relationship between PCR cycle number and amount of DNA. Wherever you capture, you will get the same difference. If it is 4-fold, it will be 4-fold at the end of 20 cycle, 25 cycle, 30 cycle, but only condition is do not do it in this region, then that will be a incorrect reaction. Okay? So, you must have an idea that within how many cycle I have to measure the product. Okay? Within how many cycle I have to measure the product. Sometime we can do some preliminary experiment to see that what is the logarithmic phase. What is the logarithmic phase, and we try to do. So, you may argue that okay, if that is the condition, why do I take a risk? I will stop at 10 cycle. Na baba, not taking risk, I will stop at 10 cycle. But then the problem is the amount has to be detectable. It should be detectable, right. For detection you can depend on machine, you can depend on your eyes, but of course nowadays sophisticated machines are there. So, we believe more those machines rather than depending on our eyes. So, a critical amount has to be reached, at the same time you should not cross this straight line phase. Two things you have to have in mind to do the gene expression analysis, right. So, in the lower graph you can see here also actually the copies are increasing, but they fall flat. Why, you know? Because it is lesser than this big number, so they looks very close, but actually they are also increasing in linear phase, but they fall flat to your eyes, or whatever detection machine you are using, it cannot make the difference. So, they fall flat, and then after 25 cycle it is increase is detected. So, it is not that here increase is not there, increase is happening, and the same kinetics increase is happening, but since my machine or my eyes cannot detect, so I will think, oh, I do not see any difference between these two bands. So, you have to wait till 25 cycle and then you see the difference, you say yes. Now I can see. Now, so detection is important. So, oh, you have run agarose gel. How do you run? You have taken your sample, you have added loading dye, fine, and then how do you visualize it? After running, you have added at some point ethidium bromide. Either you have added into the agarose gel, or you have added into the buffer, or you have done this staining after running, whatever, at some stage you might have added ethidium bromide. And you know how it works? Ethidium bromide is a dye which intercalates into the DNA, and once it forms a complex with DNA, it fluoresces. Otherwise not. Only when it complexes, it fluoresces. So, at any point of time, you can stop your PCR reaction, add ethidium bromide, do gel electrophoresis, you can see this kind of difference. In one case, the end product is more, in another case, end product is less, like that. So, you will say here my gene expression is more, here my gene expression is less. Suppose I am taking 5 different samples. So, you see here the amount will they are following the same pattern in kinetics, but different kinetics, right. So, at any point of time you can terminate the reaction, you will see the difference in the end product. Some will be having this much, some will be having this much, some will be having this much, some will be having this much. You see, you will say that here it is more, here it is less, like that. But can you say how many fold more? Your eyes cannot say. You can guess that it looks like it is double, it looks like it is 3 times, but that is not acceptable, because science cannot go by individual perception. You need to have data. So, here the machine can read the intensity of this product, how much fluorescence is there. Machine can tell you that okay, here the value is this, here the value is this, like that. So, machine will tell you that this band in agarose gel is actually 4.5x of this, means actually it is 4.5 times. Machine gives you empirical data, and then it is believable. You say okay, fine, that is fine. Okay, but that is by traditional PCR. The improvisation that have come in last 1 decade, I would say, or maybe close to a decade, we have started using real time PCR. So, the traditional PCR or the PCR that you have done for routine gel electrophoresis, that is end point PCR, means you stop the reaction and then go for detection or visualization. Whereas in real time PCR, you know what is the difference? You have been told about real time PCR. The difference is we do not stop the reaction. When the reaction is going on, we capture the reading, right. And here our intercalating dyes are a bit different. Say for example, one of the dyes that are frequently used is cyber green. It does the same thing, it intercalates with DNA. So, whenever there is PCR occurring, a DNA strand is made, cyber green is intercalated. Until and unless a DNA strand is made, it has no space to intercalate. The moment it gets a double stranded DNA synthesized, it intercalates. The moment it intercalates, fluoresce, the machine capture. So, the machine is already connected with your thermal cycler. So, it is a combination of fluorimetry, PCR machine or PCR amplification and fluorimetry simultaneously going on. Okay? And at end of every cycle, machine captures. That is the reason it is called real time. At every cycle, machine is capturing the difference between or the amount of fluorescence in one sample, another sample. Suppose you are running 10 samples. So, machine is capturing at the end of 10 cycle, it has captured the data. At the end of 11 cycle, it has captured the data, like that. Every cycle it is capturing the data, the fluorescence amount that is coming out from the reaction. So, at any point of time, you can compare and say that what is going on. But here again, you see there is a flat region, and then means this machine has also a minimum level beyond which it start detection, below which it cannot start, right. So, that again has to be taken care of, that a minimum number of cycles you have to consider before you get excited to see that what is the fold change and what is the difference in expression. That is actually called CT values. I will come into detail. Okay. So, now at the end of the reaction, say this is a experiment from my lab. You see, we have analyzed the effect of methyl jasmonate on a particular gene, that how application of methyl jasmonate affect the expression of this particular gene, and we have done this normal RT-PCR, no real time, the traditional reverse transcriptase PCR. So, we have isolated RNA from different samples, this C is control, W is some samples which are wounded, MJ is some samples which are treated with methyl jasmonate. So, you isolate mRNA from this sample, this sample, this sample, you convert them into cDNA, run the PCR, stop at a particular point, add ethidium bromide, load it on gel and see what is the difference in the end product. You see this kind of difference, and machine is quantifying this. Okay. The machine what is called not real time, I am not talking about real time now, the normal transilluminator, gel dock will read that intensity of each of these bands, and then it will quantify and show you the histogram, right, and you can easily see the difference in the gene expression. Same thing how do you do in real time PCR? Let us take a hypothetical example. So, the important thing is that now we understand the kinetics. Suppose you see this example, I have started a real time PCR reaction, and at the end of 25 cycle I found there are 1 million copies of a particular DNA, which is it is amplifying. So, at the end of 25 cycle there are 1 million copies. So, how many copies were there at the end of one cycle before, or say 24? It was exactly half of that. Fine, no problem. So, if it was 1 million copies at the end of 25 cycle, it would have been 500,000 copies at the end of 24 cycle. Similarly, 250,000 copies at the end of 23 cycle, 125,000 copies at the end of 22 cycle. Fine, you all agree? Now, if we plot this amount of DNA, which is actually the amount of fluorescence for our case, if we plot it, the number of cycle versus the amount of fluorescence, you will get this kind of curve, and as I said you that initially it will not be detected, but after a certain cycle the machine will be able to detect the fluorescence, and then there will be a straight line phase followed by a plateau phase, right. So, the critical point where the machine start detecting is called the cycle threshold, or in short form we call it CT value, right. So, for a particular sample, what do you understand? You assume that until and unless you get 1 million copies, your machine is not detecting. So, for a particular sample, if you have begun your experiment with more number of templates, you will quickly reach that 1 million copies. Quickly reach means with less number of cycle you will reach, isn't it? Compared to another case where your beginning copies are less, you will need more number of cycles. So that means what? That means lesser CT value, abundance, comparative abundance, higher CT value, less abundance, isn't it? Fine. Okay. Now say I am comparing, I am doing comparative analysis of two samples, S1 and S2, two samples. As I said, assume that in sample S1, one particular gene I have at the end of 23 cycle, I find, okay, assume that in S1 sample, you have certain number of copies, and in S2 you have 4 times more, whatever. Say in one case it is X, another case it is 4X. Okay. Here gene expression is more, so 4X, here gene expression is less, X, 4 times more. So at the end of 23 cycle, if my S1 sample produces 250,000 product, okay, 250,000 units of fluorescence, it will produce 1 million, because it has 4 times more. Same is with 24 cycle. By the time it reaches to 1 million units, this sample has already reached 1 million units at 23rd cycle. Here we reach at 25 cycle. And I said that we have a basic assumption that until and unless we reach 1 million cycle, my machine will not detect. So that is our threshold, CT. So for here, what will be the CT? CT is the number of cycle. Your threshold you have reached at 25. So here my CT is 25. Here my CT is 23. Understand? So the difference in gene expression is reflected in the difference in CT value. Difference in gene expression is reflected in difference in CT value. Now our job is that, so look at here, 2 samples. As I said to you, one case you have reached the threshold earlier, number of cycle, in another case 2 cycles later, right? Now this difference, whatever is coming in terms of CT value, you have to convert it in terms of fold difference. Okay, that's a calculation by formula. You know how, what is the amount of DNA? You know it is always 2 to the power number of cycles. That you have already learned. Learned or not? So same thing is here, since CT value is number of cycles, so mathematically we can convert, or we can find out at any point of time the amount or quantity. Here the quantity is our fluorescence, remember. DNA quantity is fluorescence. DNA quantity is more, fluorescence is more, and they are directly proportional. So using this formula, you can always convert a CT value into quantity. Do you agree? Quantity is 2 to the power CT. So if you run a RT-qPCR reaction, and if you find out the differences in CT, they can be mathematically converted into the difference of end product, that is DNA dye complex end product, and that can mathematically converted into the fold difference in the product, and that is the fold difference in the beginning point. Whatever would be the fold difference in the amount of final amount, that will be the fold difference that was there in your number of templates. Okay? So this is the whole basics about how do you find out the difference. Now there is one more term. You may raise a question, sir, always you are talking about fold difference, that it is 4-fold more here, 2-fold more here. How do I find out the absolute amount? Can't I say that at the beginning I had this nanogram of DNA? Can't I say? Yes, you can say, but then you have to prepare a standard curve. Same thing as you have done in protein estimation or for any estimation. You have to have a graph where the nanogram amount will be correlated with the fluorescent amount. Nanogram amount will be correlated. So, using that graph, any fluorescence amount can be converted into the amount of the target, absolute quantification. So, that graph has to be available. Okay? So, that graph has to be available where you will take 1 nanogram, do 23 cycle, what is the amount, 5 nanogram, do 23 cycle, what is the amount, and finally you have to plot the amount versus number of cycle and amount versus nanogram of beginning amount, and using that graph you can find out absolute. But most of the times our experiment will involve the comparative analysis where we do not bother about absolute amount. We do not need absolute amount, rather we would be needing to compare and find out circumstances where a gene expresses at higher level, etc., etc. There this kind of difference in CT value will tell you the fold change in the expression, right. Okay. So, ultimately as I showed you the formula that DNA amount is 2 to the power cycle number. So, if you take log at both sides, you get the log of DNA amount is CT at log 2. Now, it depends what is your base. If your base is 2, then log 2 to the base 2 is 1. So, that is something to do with mathematics. So, the important message is that CT value is directly proportional to the log of DNA amount, and that gives you this kind of, you see here log of DNA amount means log of copy number, and CT value if you plot you get a straight line, you get a straight line relationship. So, that is the important aspect. Okay. Now, what you are doing? You are trying to find out the difference in the number of copies of mRNA in 2 samples by doing RT and then qPCR, and you have found that whatever difference was there, that can be captured, no problem. But that difference you wanted to see, which is coming because of difference in gene expression. But how do you take care that that difference is not because of any other error? For example, you are supposed to take 1 microliter of cDNA and run the PCR. Suppose your method says that you have to take 1 microliter. You measured 1 microliter, but there is no guarantee that there is not a pipetting error. You cannot ensure that if you have pipetted 10 times, every time it will be absolutely 1 microliter, no variation. You cannot ensure that, because you pipette by your sense, you press. It is only your sense, there is a limitation of your sense. Sometime you sense it is same, but actually it is not same, isn't it? You said, sir, I pressed exactly same in every case to take 1 microliter, but there is no guarantee that you pressed exactly same. And the normal tendency is another problem, until and unless it is visible, we do not believe. That is another problem. So, suppose you are pipetting 0.6 microliter and it is supposed to be not visible, but you are not satisfied until and unless you see a liquid at the tip. So, you will keep on pressing and there is a very, very high chance that there will be pipetting error. Number one. Second, your reaction involves reverse transcription. Okay? You are doing in 10 tubes, everything you are following same. You have taken equal amount of mRNA, you have added same enzyme, you have followed same composition of the reaction, but still there is no guarantee that in all the 10 tubes your reaction efficiency was same. No guarantee. Reaction efficiency may vary, and if it varies, what will happen? Where the efficiency is more, it will produce more cDNA from a particular amount of mRNA. Suppose hypothetically, in every case I have 100 copies of mRNA of gene A, and in one tube my efficiency is 100%. So, I will get 100 copies converted into cDNA. In another case my efficiency is only 50%. So, there also it was 100, but it came as 50, and when I do all the things I conclude this was double in expression, but that was not true. This difference has come because of the difference in reverse transcription efficiency. Same could happen in your RNA isolation also. You have isolated RNA for 10 different samples. So, there is no guarantee that all the samples is having same level of purity. That in turn will affect your efficiency of reverse transcription, isn't it? So, all these are the source of errors. Sample to sample variation, it could be variation in RNA integrity. You have equal amount of RNA, but in some sample it has been degraded because of some problem. In other cases it is perfectly fine. So, you can have difference in RNA integrity. You can have difference in reverse transcription efficiency, and you can have difference in measuring the cDNA while setting up the qPCR reaction. But do you want all these differences to be captured? No, your ultimate purpose is to capture the difference which has come because of gene expression. So, you have to nullify all these differences. That is called normalization process. Normalization process helps you to nullify all these differences and make sure that whatever difference you get, it is only because of gene expression, because all other possible sources of variations have been nullified. Okay? How can you do that? The basic is, what we do, we parallelly run the kinetics of another gene which is supposed not to vary across my sample. Do you understand? Now it will depend. Suppose you are doing a gene expression study between a heat treated sample and a control. Control means untreated sample, and you wanted to see whether my heat treatment increases the expression of gene A or not, and if it increases, how many fold it increases. So, what we will do, we will take another gene, we will take means we have to get primers only, that is it. We will consider parallelly to study another gene which is not supposed to show any variation because of this treatment. Understand? We say housekeeping genes, but I say there is nothing like housekeeping genes, isn't it? Actually, you have to take a gene which is suppose not to show any variation. Traditionally we say housekeeping gene because there are certain genes which most probably would not show variation, but until and unless you check, there is no guarantee that whatever so called housekeeping genes you have considered that does not show variation. There is no guarantee. Okay. But let us first understand the basic. Basic is what we do, we take a housekeeping gene with the assumption that it will not show any variation in gene expression. Now we do the same kinetics, whatever we are doing for gene A, we will do for this housekeeping gene also. Okay? Then we will compare that whatever difference has come in the expression of housekeeping gene or in the end product of housekeeping genes. That we consider that that difference is because of all these factors. Because our primary assumption is there is no difference in gene expression of the housekeeping gene. So, whatever difference you get in the kinetics or in the end product of the housekeeping genes in your two samples, that you consider because of all these things. So, the same amount of difference we subtract from the difference that we have got for our target gene. Understand? Again in simpler terms, suppose your housekeeping gene has shown two-fold difference. Okay? Means in one case it is say 1x, another it is gone 2x. A two-fold change has occurred and you have considered that okay, that two-fold difference is because of all these unwanted things. And now if your sample shows 8-fold difference, you will subtract two-fold. You will say no, 2 out of 8-fold, 2-fold is because of unwanted things. So, you will say no, 6-fold I accept. So, this is normalization. In simple way, this is normalization, but again there are mathematical formulas to do that. Okay? Mathematical formulas to do that. Now, in your experiment, you have to decide that which gene will be good for housekeeping genes, and since you are from entomology background, I saw that there are various genes that are frequently used as housekeeping genes for experiments on insects. Okay? These are some of the genes like 18S rRNA, actin 1, elongation factor 1A, and so on, and so you can always find in literature, there is no end to it, and again some more I found that so many genes can be used as housekeeping genes. But remember, if you do not see a literature which directly says suitability of a particular gene to be used as housekeeping gene, your responsibility lies in assessing the suitability of that gene. What do you mean by assessing suitability? You have to ensure that whatever you are studying, this gene has no effect. Suppose you are studying effect of starvation of an insect, and you saw in the literature, you see that for your insect species there is, I mean, no paper says that it is suitable. So, your responsibility lies to see whether if you have a plan to use say GAPDH or say elongation factor 1A, your responsibility lies to see that expression of elongation factor 1A is not affected by starvation treatment. Understand? Or many a times what people do, instead of one, they take 3-4 housekeeping genes in doing an experiment. Advantage is that even if there is some variation or some deterrence in 100% suitability of a housekeeping genes, if you consider many, the error will be further reduced. So, you will be more close to appropriate interpretation of the data. Understand? So, already I have told you how the normalization is done. You have to parallelly run the same qPCR or RT-qPCR for the housekeeping gene, or you have prepared the cDNA, on same cDNA you do qPCR with your gene specific primer as well as in another tube with your housekeeping gene specific primer. So, it will be 2 tubes parallelly for each technical replicate, 2 tubes. Same cDNA, in one case I have added gene specific primer, in another tube I have added housekeeping primer, and then I have run. Then I will study first the housekeeping end product of my housekeeping genes between these two samples. Is there any difference? or I will study the CT value in if we do qPCR. If we do qPCR, I will see that is there any difference in CT value. If you see there is no difference in CT value, fine, but if you see there is a slight difference, you have to nullify that from your experimental data. And in qPCR, I have just given you one example. It is again accessed from web resources. In qPCR, how we do? So, we find out the CT, say here, sorry. In this example, suppose in an experiment we are trying to find out the effect of drug treatment on a particular gene CMYC. Okay. So, I have 4 samples untreated, treatment A, treatment B, treatment C. I have calculated the CT value. So, these are my 4 tubes. Okay. Control means untreated, tube 1, tube 2, tube 3, tube 4. I have taken one housekeeping genes, again tube 1, tube 2, tube 3, tube 4. So, total 8 tubes for one technical replicate. If you want to replicate, again another 8 tubes. One more replicate, another 8 tubes, fine. Then you take the, so you run qPCR, you prepare cDNA, run qPCR, machine will show CT value in each tube. In each tube, you will get CT value, machine will simply show you, you need not to do anything. So, now depending on the replicates, you have to average them, that is the reason we take replicates. So, you average them. So, you find out the CT value of untreated, average value plus minus something, CT value of treatment A, CT value of treatment B, CT value of treatment C. Okay. Same for the housekeeping gene, CT value of untreated and then CT value under treatments. Now, what we do in case of delta CT method, this is a one particular method of normalization, you can get a few more methods also. So, in delta CT method, first you find out the delta CT of your respective housekeeping genes. Okay. Delta CT means how it is it is not visible to myself. So, what is the difference in CT value between your sample and the housekeeping gene that you note down or machine will calculate, and the same way you find out the differences, fine. Why you have found out differences? You are actually nullifying, I said to you, normalization. So, this is the process of normalization, you are subtracting in every case. Okay. And then if you are interested to see the effect of treatment, there will be again subtraction between treatment and untreated, understand. So, like say I want to find out the difference, I want to find out the effect of treatment A. So, delta this delta CT minus this, treatment B delta CT of this minus untreated, treatment C delta CT of this minus untreated. So, it will be delta delta CT, that is the reason it is called delta delta CT method. Okay. So, first delta is to normalize the sample, next delta is to find out the effect of treatment on gene expression. Is that clear? If you have any doubt, please raise. And then once you find out the difference in CT, that can be converted into difference in amount. You remember the formula? DNA amount 2 to the power CT value. So, same thing, DNA amount is equal to 2 to the power delta delta CT. So, using this formula one can convert the CT into the difference in DNA amount, and that is your fold change. Difference in end product DNA amount is the difference in the beginning template amount. So, that is your gene expression level of. but all these things will be done by machine. But until and unless you are clear in basics, you will not be able to properly handle the machine. Machine does not have its own intelligence, you have to dictate it, is not it? So, you have to understand that what you got to do, and accordingly you ask machine to calculate all the values and show you. Okay. Now, as I said to you that again, I mean with due course of time, many literatures they indicate that none of the gene is actually universal which can be used as housekeeping gene. One has to really find out whether or ensure that there is no effect of that particular treatment that you are experimenting on that particular housekeeping gene that you have decided to use. Okay. Therefore, none of the reference gene is really can be universally used. One has to do validation. One has to do validation. So, the validation itself is another piece of work. You have to consider several candidate reference genes, you have to study their expression under that particular treatment regime. Suppose you are working on the starvation effect on a particular insect species. So, starvation for 4 hours, starvation for 8 hours, starvation for 24 hours, you have 10 samples. So, you take set of candidate genes, you study their expression in that regime of starvation and see do their expression changes. If not, then that is fine, but if it is changing, then you have to consider another reference genes. There are many statistical algorithms which are available to find out that which is a good reference gene, which is a good normalizer. Statistical algorithms like these are very popularly used statistical algorithms like G-norm, norm finder, best keeper, delta CT. There are many more. There are many more, and again it's developing area. People are proposing new and new method. I will not go into detail because they are basically statistical algorithms. So, until and unless you do practical, it will not make much sense in talking on them. But just to tell you one of these methods, say for example G-norm, how it works. What it does, the algorithm is written in such a way that when you study a set of candidate genes, it takes 2 at a time. So, I have say HK1, HK2, HK means housekeeping. So, HK1, HK2, HK3, HK4, HK5 like that, HK10. I have 10 reference gene and I want to study that which one is really stable in this regime of starvation, that is my experiment. So, I will do all this RT, Q, PCR, I will record their CT values, and then what this G-norm will do, it will do pairwise comparison. Pairwise comparison means it will take HK1 and HK2 at a time, compare, find out which is better, then take HK3, compare which one is better, exclude the other one. Like that it will do pairwise comparison, it will see where the variation is less, and then it will do that. And in a situation, suppose you could find out more than one reference genes which are seemingly suitable but not absolutely suitable. You understand? Which are close to be very good but not absolutely fine. You can use more than one reference gene in your experiment. So, the simple change that we do, whatever CT value we record, suppose in this case I am considering 3 reference gene at a time, although it is showing 5, but for simplicity, suppose I am considering 3 reference gene at a time. So, I will take CT values of all the reference genes and then convert them into corresponding quantities by that formula 2 to the power CT values, and then I will take geometric mean. Instead of taking one value, I will take geometric mean. Okay? Geometric mean of these 3 I will take, geometric mean of these 3. So, and then I will call it as normalization factor. So, it is not of one gene, but it is a normalization factor where I have considered multiple reference gene and have taken mean of that. Why geometric mean? Because we are talking in terms of fold change, we are not talking in terms of absolute amount. Had it been absolute amount, you could have think of arithmetic mean, is not it? Since it is fold change matter, so we are taking geometric mean and then we are using it as normalization factor, and then the same thing we are doing as you have seen in the previous slides. Okay. So, methods are available. Important thing is to know, if you encounter a situation where you got to find out a suitable normalizer, then its experiment has to be done. Okay. You can also see one of the papers that have been published by one of my students, where we have done similar kind of analysis to find out good reference genes that can be used for that can be used as a normalizer in case of mustard aphid. So, we have taken different regime of treatment, starvation, heat treatment, developmental stages, different developmental stages, and then we have taken set of reference gene, 10, 12 reference gene. We have analyzed and tried to show how they are varying under the different regimes of treatment, and finally, we have recommended that how one can take care to identify a good normalization process in doing gene expression study in mustard aphid. So, you can go through this paper. This is open access. If this is open access, you can see how it has been done. I am not going into detail in that. So, I conclude with this, but I will be very happy if you have any questions, you can raise, and I would be very happy to clarify. Yes.
Sir, actually in one of your slides, the concentration of the tablet for the QRT PCR. Sir, in one of your slides that was mentioned, concentration of the QRT PCR less than 1000 nanograms.
Less than 1000 nano. Not nanogram, they are units of fluorescence. That I was talking that what is CT value, right? 1 million you are talking, no? 1 into 10 raised to the power 6.
Quantity or the concentration of the template we should take in the QPCR.
No. Let me go back. Which slide you can point me? You can tell me which one, I will stop. This one? This, anything here? Table. This one? Oh, acha, okay. You are asking this. No, this is just arbitrary. See, I have just taken this is a hypothetical. It is nothing like that. It has to be less than 1 nanogram. No. I am sorry for that. You have to take the cDNA template, and this is my purpose was just to make sure that you understand what are the things in a PCR reaction. You don't go by the values.
Like in the previous lectures, I got to know like less than 100 nanograms is like sufficient. But for the low expressing gene.
Yes, exactly. You are very much right. We cannot arbitrarily say that it has to be less than 1000, because here we are talking about the cDNA amount, isn't it? So, always what will be the target mRNA present in that amount of total cDNA, that always matter. Yes, you are right.
Just can we go with like quantify the total RNA and the cDNA? We can abide by the readings of the others. Like what is the quantity?
Yeah. See, as I said, that one of the source of variation is your you cannot afford to have differential amount of mRNA in different reaction, isn't it? So, those precautionary steps are already there. These are standard practices that when your purpose is comparative gene study of comparative gene expression, you have to make sure that you are taking equal amount of RNA. You have to make sure. And how do you make sure that you are taking equal amount of RNA? You have to use spectrophotometer or nanodrop to quantify each sample, isn't it? Remember, once we say equal amount of RNA, it is not that 2 microliter in each case. 2 microliter has no meaning.
It's a volume. It's just a volume.
It's just a volume, isn't it? You have to go by concentration. What is the amount that you have to take in each case? So that is a part of your standard method, but still, that's what I was trying to explain. Suppose I have taken equal amount of mRNA, RNA or mRNA, whatever. There is no guarantee that in all the tubes RT reaction is equally efficient. Even if you measure purity by spectrophotometer, there is no guarantee that the purity is equal, because in spectrophotometer mostly you measure impurity in terms of presence of protein. So if there is difference in purity, that will affect the difference in RT reaction, and that will be your unwanted part of variation. So that is the need of using normalizer. See, suppose you are using a housekeeping gene as normalizer, and then you are seeing that your across the samples the CT value of housekeeping gene is almost same. That indicates that your experiment has been done in a very good way. And suppose there is already so much of fluctuation, that indicates that all these set of steps you could not do properly, there is already so much of variation, isn't it? So that is there, and that is the need of parallelly running the kinetics, because other things you cannot ensure. You have to take care that equal amount of pipetting, everything is same, but you still cannot ensure. Therefore, you are depending on kinetics of a molecular reaction, and then accordingly normalizing the samples. Melt curve, okay, significance of melt curve. See, had I known, I could have taken that also. Have you seen how a melt curve looks like? Right? So, what we do in preparing a melt curve, whatever product we have got at the end of say qPCR or qPCR, mostly qPCR. Now we try to melt them. How do you melt a DNA strand? Melt means you denature them. So we subject them in a stepwise manner to higher temperature, incremental dose. Fine. So what will happen? They will, two strands will dissociate and fluorescent will go, because it is the dye that you are using, say cyber green, it is an intercalating agent. So if two strands are separated, the fluorescence will be lost, right. So in this way we slowly increase the temperature to see that whatever product you have got, they show the same peak denaturation, because for a particular DNA the peak denaturation, peak you understand, you increase the temperature, they will be denatured, but at a particular temperature maximum molecule will be denatured. So that single peak indicates that in your tube whatever product has come, they are one type. That indicates the specificity. If your product is one type, means you have amplified only one particular region. So melt curve analysis is done to ensure that whatever amplification has happened, it was specific as per the desired amplification. So desired target has amplified only, and if it is not, if there is some non-specific amplification, you will get more than one melt curve. Is that okay for you to clear it out? Thank you. Then
Now we come to the thank Dr. Ramcharan Bhattacharya for offering a very good and excellent lecture. This is